Welcome to the cutting edge of RNA therapeutics. I'm your host, Christopher Brandon. I'm an assistant professor at the Department of Cardiovascular Sciences and the Center for RNA Therapeutics at Houston Methodist. And I'm joined by my co-host and colleague, Daniel Kiss, who's an associate professor also at the Department of Cardiovascular Sciences and Center for RNA Therapeutics. And he'll be introducing our guest today, Dr. Alexander Wesselhoff.
Thanks a lot, Chris. It's my pleasure to introduce Dr. Alexander Wesselhoff, who is the Director of RNA Therapeutics at the Cell and Gene Therapy Institute at Mass General Brigham Hospital. And he's also an instructor at the Division of Engineering and Medicine at Harvard Medical School. He earned his PhD at MIT in Dan Anderson's lab, where he invented a groundbreaking RNA circularization methodology and that has helped establish synthetic circular RNA as a research and therapeutic platform. During his postdoc, he pioneered scalable circular RNA engineering, manufacturing, and delivery strategies that address key issues in RNA therapeutics, like the stability, immunogenicity, and payload size challenges of messenger RNAs. After his postdoc, he co-founded Orna Therapeutics and headed its RNA research and development group, advancing circular RNA medicines toward the clinic. He currently oversees the RNA Therapeutics Core and their attempt to democratize circular RNA research and developing next-generation RNA medicine platforms for diverse therapeutic areas and for many different clients. And today he'll discuss circular RNA, engineering, manufacturing, and medicine. And to introduce our panelists, I have two members of my lab, Dr. Katharina Castillo and Dr. Nada Bajar. They're both research associates in my team.
And I guess before we get started on your presentation, Alex, I wanted to start a brief discussion about the future of circular RNAs. I guess one question I had is kind of like, in the next few years, what are some of the underexplored applications, I guess, and technical updates that you imagine or are excited about for circular RNAs that are sort of underrepresented in the field right now?
Yeah, it's a great question. First of all, thank you very much for having me here today, and I'm always happy to talk about circular RNA. With respect to the development of the fields, I think circular RNA is a relatively young field, especially when you look at the development path of mRNA over time. It took decades of really foundational research to get to the point where we were able to make an mRNA vaccine, which is probably one of the simplest forms of RNA medicine that you could make with mRNA. Not to discount the complexity of how complicated and difficult it is to make an mRNA vaccine. But when we think about transitioning the RNA field from this sort of starter set of applications in vaccines into more complicated biology, such as addressing autoimmunity or cancer, these problems start to become a lot more complicated, and the requirements on the technology become a lot higher. And that's where I see circular RNA as starting to have advantages over mRNA, where you get maybe more protein per molecule, so you don't have to deliver as much, so you have a better therapeutic index. Over time, I would say that circular RNA may even become a modality of choice for coding RNA therapeutics. In terms of the really key innovations that I would say would be coming up on the technology front over the next few years, delivery technology is seeing a huge amount of activity and innovation. And I think that the delivery problem is basically the problem to solve right now. And the cargo can reduce the needs on the delivery system by being better, but fundamentally getting to the tissue types where the disease is active is the key problem in the space of RNA therapeutics right now, and that's where most of the key investments are being made.
Do you feel like circular RNA is going to face pretty similar delivery problems to linear mRNA, or are they going to diverge a little bit in some way?
Yeah, so I would say the problems are very similar, although circular RNA has the advantage of being a little bit more stable, a little bit easier to make, a little bit cheaper. And so those enhanced properties can open doors that are shut to mRNA because of its short half-life. and other drawbacks. So the fundamental delivery challenge of getting to a cell type of interest is going to be basically the same as mRNA, but it's probably going to be more achievable the better your cargo is.
All right, yeah, I guess I'll let you go and get into your presentation when you're ready.
I'll jump right in. So it's my pleasure to be here to talk about the work that I've been engaged in the field of circular RNA for the last almost 10 years now. Not quite, but almost 10 years. And I've been working on circular RNA since my PhD, where we started out from basically from scratch. The field didn't really exist back then. And it's been an amazing journey to see how far circular RNA has come as a technology, and how close it is to clinics even today, where several companies are moving into clinical trials in the very near future. So without further ado, I'm going to get into the talk here and discuss at a high-level overview some of the work that has been done on circular RNA sequence engineering, the ability to manufacture circular RNAs, and some of the very early medical applications of circular RNA. So RNA has really exploded in the last few years as both a research tool with control over dosing, redosability, ease of multiplexing, relatively low toxicity, and a high degree of control over the composition of matter, and pretty easy developability compared to other platforms like antibodies, for example. And on the therapeutic side, we've seen approvals of the first mRNA vaccines, and we have cancer vaccines in clinical trials, rare genetic disease mRNAs in clinical trials. And so we've seen a lot of clinical activity out of mRNA in the last few years that has really propelled a new wave of RNA technology development. Most of this has happened with mRNA, which has been in development for quite some time. But mRNA has, while it has all these advantages in terms of developability and dosing and design, it does have drawbacks, with the main drawback being that it's a highly unstable molecule, and it's pretty expensive to make as well, due to the cap and the tail. So back in 2015, in the Anderson lab, while I was at MIT, we were looking at this problem of mRNA instability. And one of the thoughts that we had was, okay, well, mRNAs are super unstable. They only last for a few hours. We know that this degradation or the instability of the mRNA is caused primarily by exonucleases. You can see that these different pathways of mRNA decay that have been studied over the last few decades. And on the right, this is this massive exosome complex that's responsible for most of the turnover of RNA within cells. So if these exonucleases, which trim the RNAs from the ends, are responsible for most of the instability, is there a way that we can prevent exonuclease mediated decay of the mRNA? And one of the most obvious and clear solutions to that is just simply take away the ends. If you don't have any ends, then you can't be targeted by these exonucleases. So that's really where the concept of circular RNA as a more stable form of mRNA arose during that time. And the thinking was that if we could circularize the molecule, we could make it more stable. And by making it more stable, we get more protein produced by the RNA molecules per RNA molecule. We get a dosing advantage, and then we get a better therapeutic index, which opens up completely new avenues of medicine that mRNA can't really reach because of its instability. So we started with the question, okay, how can we make circular RNA? And I'm going to discuss a little bit about the engineering processes that we went through to be able to make these things in the first place. So back in 2015, the state of the field was pretty sparse. From the synthesis perspective of being able to make circular RNAs, people had shown that you can make small non-coding circular RNAs that are maybe 100 nucleotides. They can't encode a protein. They're not big enough to be particularly useful, but they could be inefficiently made. But big protein coding circular RNAs, we hadn't seen any examples of those being made, and certainly not efficiently. no one had shown that synthetic circRNAs could be translated into proteins, so that was still an open question mark. And then from a therapeutic perspective, of course, because people didn't really know how to make them and didn't know if they could encode proteins, there was no therapeutic field for circular RNAs whatsoever. So the first thing we did was we designed this system to produce large circular RNAs in a reliable and repeatable way. And what we did was we took a group 1 intron, which this has been studied, by other groups in the past and even used to generate very small circular RNAs in the past. And we split it in half, took the second half, put it in front of the first half, and then inserted a sequence of interest that had an internal ribosome entry site and a coding sequence in between those group 1 intron fragments. And the idea is that this group 1 intron would basically catalyze a backsplicing process to create circular RNA while these molecules are being synthesized in vitro. This initial system didn't work for large circular RNAs, and that's probably why there had been limited success in the field before. But long story short, we engineered in a bunch of accessory or auxiliary sequences into this molecule to facilitate proper folding and activity. And after this engineering process, we found that we were able to circularize lots of different types of large circular RNAs, ranging from the smaller Gaussioluciferase erythropoietin circular RNAs to the larger Cas9 circular RNAs. So this was the first major step that we made towards being able to make protein coding circular RNAs efficiently in a way that was potentially scalable enough to become therapeutically viable. The next thing we looked at was translating protein from circular RNAs. And normally mRNAs have this cap structure shown on the right, where proteins in the cell will attach to the cap structure and then initiate translation. This cap structure is located on the 5' end of the mRNA. And in a circular RNA, we don't have any ends, so we can't use a cap. So what we did was we tried a solution from viruses, which is an internal ribosome entry site. And we took this internal ribosome entry site, which is this relatively big RNA structure. It doesn't encode any proteins, but it folds into a way that mimics a cap structure in some ways. And we put it into the circular RNA. And we found that indeed, when we did this, we could actually drive protein translation from circular RNA. And so this was the first time we were able to see protein being generated from these synthetic circular RNAs in actual living cells. When we look at IRESs, we see that they have much greater diversity than caps in general. And there have been a multitude of caps that have been explored for mRNA, but generally the sequence space is pretty limited. So you have basically 3 bases with different methylation sites that you can add, where you methylate it and makes it a cap 1 versus a cap 2 versus a cap 0. And the key decisions are the nucleotide identity next to the cap. So not a ton of space to screen through. But with IRESs, IRESs are huge. They're like 500, 600 nucleotides long. So you can have almost infinite combinations of IRES elements. So that was the first sort of functional diversity problem we were running into, was how do we screen through all this space? So we did a preliminary mini screen looking at different IRES elements, and we found that this handful of 6 different IRES elements showed completely different levels of protein translation, indicating that the sequence identity of the IRES mattered a lot, and there was a lot of diversity in terms of function. And like with caps, there are a few different types of IRESs, but even within IRES types, for example, this is an enterovirus IRES comparison shown here, you can see that there's a huge amount of sequence diversity. So when we were looking at the potential diversity of IRES elements, again, it's almost infinite. Even closely related IRESs have very significant differences in sequence, and those differences in sequence appear to translate into differences in function, shown on the left, comparing poliovirus versus CVB3 IRES elements. CVB3 is clearly more potent in this context than PV. So early on, and this is work that I led while at Orna Therapeutics, we decided to basically screen through all known natural IRES elements, because that was a pool of IRES elements that was just there, sitting there, that we could characterize computationally, and then we could screen through them to find the best ones that nature has selected over a long process of evolution. So we computationally identified about 3,000 candidates that were down selected from a larger pool of all known potential IRES candidates. We synthesized DNA, we made purified circular RNA, we formulated into lipid nanoparticles, and then we tested in 3 different primary human cell types. Those cell types were T cells, myotubes, and hepatocytes. And what we found was sort of mimicking our mini screen, which is a massive diversity of IRES elements, giving rise to a massive diversity in function. And we found that IRES elements that are commonly used in the academic sphere for a variety of purposes, like this cloning vector EMCV here, was actually not a very good IRES. And compared to some of the IRESs that we were identifying in these primary human cells, we were finding IRESs that were 40 times better than the cloning vector EMCV IRES. And so this was quite exciting and really is the first major characterization of these IRES elements in such a comprehensive way to date. We also looked at how structure and sequence were related to function. So on the left, these are the IRES elements are clustered by structure, and they're colored by potency. So you can see that some of these clusters light up more than others. So clearly there are some structural similarities between the IRESs that work. When we look on the right, there's a lot going on in this one. I'm not going to cover it in detail, but basically it's showing the relationship between the structures of the IRES elements and their function in the 3 different cell types, which is delineated by the 3 purple rings, and then the taxonomy, which is the outer ring. And so a lot of the IRESs that worked were related in terms of structure and taxonomy, and they sort of clustered in a few distinct clusters. All right, now I'm going to talk about manufacturing, which is of course critical for generating a drug substance that can eventually be commercialized. As many sort of RNA enthusiasts are aware, the in vitro transcription reactions that people use to make RNA produce inflammatory double-stranded RNA as a byproduct. And this double-stranded RNA increases the immune response to the material, which lowers the potency and expression of the material. And this is a huge problem. And in my opinion, it's basically why nucleoside modifications have been employed for mRNA, because they essentially prevent the immune response from happening, even if you have a little bit of double-stranded RNA in your preparation. So you don't need to purify it quite as much. So we wanted to look at whether or not this was important for circular RNA. And unfortunately, with circular RNA, you can't really modify it. So you have to go the purification route to remove that double-stranded RNA. And we developed this sort of long and onerous process of purification where we would do an RNA-SAR treatment, then HPLC, and then phosphatase on top of each other to get highly purified circular RNA. And we found that that was tightly correlated with the function of the circular RNA and the cytokine response to circular RNA. And we found that in RLR active cells, there was essentially no cytokine response to purified material. In TLR responsive cells, TLR 3, 7, and 8, we saw that purified circular RNA looked basically the same as modified mRNA without the need for modifications. Of course, this process that we developed, which is briefly shown at the top here, was extremely onerous, and you only got a little bit of RNA out in the end. So at Orna, we spent a lot of time making this process very quick and clean, and we were able to develop a highly efficient process where we could circularize the material during the in vitro transcription reaction and then go straight into an improved purification process that yielded purified circular RNA that looks like the chromatogram on the bottom left middle here, where we see highly purified material. And when we test these materials in vivo, what we see is that the highly purified material denoted ultra pure here actually stimulates an even lower TNF alpha response than modified mRNA. And so this really goes to show that the purification is super important, but once you get it down, you don't really need the modifications in vivo, and you can still have something that is potent and scalable with the right process. And so to sort of capture a lot of these innovations and the movement of the field in improving our ability to make, design, and test circular RNAs. I've led the opening of the RNA Therapeutics core at Mass General Brigham with the goal of democratizing the circular RNA technology. And the core manufactures stock circular RNAs, custom circular RNAs. It can formulate circular RNA into lipid nanoparticles and produce almost any scale of research-grade material necessary. We're also working to open a GMP facility, which is currently in process to be able to support manufacturing of clinical-grade circular RNA and lipid nanoparticle formulated circular RNA. So we've got the email in the bottom left there and the website in the bottom left as well. Feel free to reach out. So here's just some examples of the stuff we're making at the RNA Therapeutics core. So on the left is a 2.5 kilobase circular RNA purity. It's pretty good purity. The cytokine response is essentially non-existent compared to this control mRNA in these cells that express the dsRNA receptors. And then when we look at the stability and potency of the circular RNA, we can see that the material actually outperforms mRNA quite a bit. So on the left, you can see EGFP expression. So this is just a reporter fluorescent protein. The protein accumulates over the course of the 6-day experiment, whereas the mRNA starts at day 1. It starts relatively close to circular RNA, but it's not really producing much material after day 1. And then you can see the same effect on the right, looking at these two different cell types when it's formulated in a lipid nanoparticle using a different protein, which is much more unstable. You can see that the circular RNA produces a lot of protein for a longer duration than mRNA, which sort of fulfills the original vision that we had for circular RNA when we started working on it back in 2015. We're also developing new technologies and improvements to continue to push forward the field. We have developed a codon optimization algorithm that improves the stability without compromising on expression. Also, it improves manufacturability of circular RNA specifically. We're developing new circularization systems to improve circularization efficiency for really big stuff. And we're developing some unique biodegradable ionizable lipids. So I'm going to talk a little bit about the medical applications of circular RNA and where people have been using circular RNA. Circular RNA itself is a platform technology. It's very versatile, and compared to other modalities like plasmid, AAV, or lentivirus, or mRNA, it has different advantages and drawbacks. And compared to some of the DNA methods like virus or plasmid, circular RNA is not going to last as long. It's not like hyper stable. It's not going to be there for months or years. It's going to be there for days instead of hours compared to mRNA. But for any sort of transient application where you want a burst of expression for like a week and then it goes away, circular RNA tends to have a lot of advantages over the existing mRNA technology. One of the areas we've looked into using circular RNA is, of course, vaccines. So we did a little bit of work on a spike vaccine for COVID and showed that we could produce, this is the crude raw material from the IVT shown in the chromatogram in the center, showing about 68 to 70% circularization efficiency for this relatively large RNA. And in mice, we were getting pretty good binding antibodies upon vaccination with circRNA. We've also looked into it as a rare genetic disease platform, and here's just one example looking at full-length dystrophin. And I show this example because full-length dystrophin, that protein is massive, and the RNA size here for the full-length dystrophin is about 12 kilobases, which I'd say is pretty much the upper limit of RNA therapeutics. And we showed that we could actually make these circular RNAs, and we were able to get expression in cell-free assays of the full-length protein, as well as in primary human myotubes. And finally, this is probably where most activity is happening in the industry right now, looking at in vivo CAR T cell therapy, basically transforming CAR T cell therapy from a personalized cell therapy into an off-the-shelf medicine to really allow people to access it, expand the access to this revolutionary CAR T drug. What we do is we basically take the chimeric antigen receptor, we encode it within a circular RNA molecule. We put that molecule inside a lipid nanoparticle. The lipid nanoparticle brings it to a T cell and transiently converts the T cell into a CAR T cell, which then kills the tumor cells, and then it all goes away and back to normal. And you can redose, as much as you need as necessary. And on the right, you can see some data looking at this in vivo CAR T circular RNA drug that is dosed 4 times, and you can see that the mice start with significant diffuse lymphoma, which is shown in the blue color, and then after a couple of doses, it's completely gone, whereas the control group unfortunately succumbs to the cancer. So this is a very exciting new technology application that I think there's probably going to be clinical success in this space in the coming years. So to summarize, we started with a dream of a better circular RNA. We figured out how to make big circular RNAs. We figured out how to express protein from them. We did a lot of optimization to get them to express more protein than mRNAs. And we figured out how to manufacture them. And we started to explore different medical applications of these materials. And so it's been an incredible journey to see the development of these materials over time. And I think the future is bright for circular RNA as innovators from around the world continue to push the technology forward. So with that, I'd love to thank my team at the RNA Therapeutics Core and within my research lab at the Gene and Cell Therapy Institute of Mass General Brigham, and the many collaborators that I've worked with on various circular RNA projects.
Thanks, Alex. That was a really great talk, really exciting stuff. I have a lot of questions, but I wanted to sort of open the floor up for our guest discussants, Kat and Nada, and see if they had any questions for you off the bat here.
Yes, thank you for your talk. So my question is on the 2-step process for IVT and circularization. So is there a limit to the size of template and then the final circle that you can use? I assume that with longer circles, the circularization efficiency is also affected in some way.
Yes, absolutely. The larger circular RNAs are more challenging to make. At some point, it's less so the circularization process and more the degradation of the large RNA during the process that becomes the problem. Mm-hmm. You see this with mRNA or linear RNA too. Once you get about 7, 8, 9 kilobases, you're starting to get a lot of nicking in the backbone of the RNA. It's hard to tell with linear RNA because it sort of all smears together. But the nicking starts to dominate after a certain point.
I do have a question. Very, very nice talk. First question is, so when you tested all of these irises, have you observed any difference in the circularization efficiency? The way I see the pipeline is that there isn't really, I don't know if it's not shown only, but there isn't a check on the circularization, I guess, before testing on their effect. So is there an effect on the iris on the circularization efficiency?
Yes, the iris can definitely affect the circularization efficiency. We designed the backbone to be as permissive as possible for swapping different things in, but it can certainly happen. We did have a purification step before testing those. So generally a disruption to circularization efficiency would be reflected in lower yield. So they should be still all pretty much on the same level in terms of purity.
And my other question is, there's some literature saying that some scarless circular RNA produced less immunogenicity. What's your opinion about that?
Yeah, so I think because circular RNA doesn't use modifications, the bar for purification is very high. Like actually we published on this in 2019, femtograms of dsRNA contaminants will trigger immune responses. And so that introduces a lot of variability into experiments. when it may look perfectly clean on a chromatogram, but you're not going to be able to see femtograms of dsRNA on a chromatogram. There's just not enough of that on there. So most of the field of immunogenicity of circular RNA, then that entire body of literature, is looking at dsRNA contaminants and not necessarily the circular RNA itself. And the scar that remains behind in the circular RNA is it doesn't do anything. It's basically a UTR spacer that doesn't contribute to the function of the material. But you actually kind of want some spacer in there anyway to help things fold correctly. So I'd say it's harmless at worst and maybe a little bit helpful at best.
I guess, are you surprised that all the circular RNAs that you work with are so low in their immunogenicity? I mean, would you expect some of them to form shapes that would set off some of these double-stranded RNA sensors in cells?
That's definitely something I was worried about, especially in the early days. Taking an IRES element from a virus, that thing is known to be highly structured. And if you form a lot of dsRNA via the IRES, are you going to stimulate an immune response? I think luckily IRESs have evolved to not stimulate immune responses through their IRES. So I have never seen a case where an IRES is immunostimulatory, but that's not something that we've actively screened for in these really large screens. So I don't want to rule it out as a possibility, but I have never seen it happen.
Okay. One question I had is, are you guys looking at sort of switchable elements or structural elements that can be activated so that you can get context-specific or cell type-specific activation and expression from these things?
Yeah, so cell type-specific expression is like a huge area of development. I think there's some great preliminary work done. I think Toeholz, I forget the group that did it, I think they were at MIT, looking at switchable IRESs using these autoinhibitory loops that then get de-repressed based on the nucleic acid environment of the circular RNA. So yes, definitely this is of interest. I think my main hesitation with that is that typically it's kind of hard to engineer one that is very robust. And you're always going to have some leaky expression. You're always going to have probably some reduced overall peak expression. So you're going to compromise on the potency of the molecule if you're doing so. So it becomes a trade-off of, okay, do I really need to be in just this cell type, or can I afford a little bit of off-target protein expression and have a more potent molecule?
Thanks again for a great talk. If groups out there are looking to your circular RNA technology and adopting it in their lab, aside from contacting you at the website and ordering it from you, what tips would you give to someone that's looking to set up your circular RNA methodology in their lab for research use?
Yeah, what tips? So I would probably start with published sequences. I think a lot of things have already been optimized, and it's kind of a lot of work versus starting from scratch. There are some pretty good published sequences out there at this point that form a good baseline for being able to make circular RNA at a small scale. And you don't necessarily need to be able to make it like really large scale for research purposes. So a lot of the existing methods are like pretty robust if you follow them closely for generating purified circular RNA. I think there is some activity, like people are moving towards new purification methods like chromatography, which I think have a lot of promise for smaller scale reactions. Overall, I think the main pitfall with circular RNA research and academic research is purity and making sure that you are testing the immunogenicity of what you really expect to be testing, and making sure that any immunogenicity can compromise the function. So the quality of the material is a significant pitfall, and if any effort should be made, it should be made there.
Yeah, and to follow up on that, would you recommend a particular benchmarking assay or two for groups that are looking to make their own circles?
Yeah, so in terms of benchmarking, like material quality, I would generally benchmark against a commercially available modified mRNA because it's something that everyone can buy. Even though it's mRNA circRNA, everyone can buy it, so everyone knows how it performs. And a lot of groups don't they'll benchmark against something they make themselves, or they'll benchmark against something they read from a patent and maybe they didn't quite read it right or something. I think the best benchmark is always going to be, buy commercially available modified mRNA off the shelf stock material if possible, and benchmark against that.
I had a question about your codon optimality formula for the circular RNAs. Presumably there's still not a non-existent stability problem, and it seems like you've improved stability with your codon optimality. Can you describe a little bit of how mechanistically that works? Does it have to do with ribosome occupancy or collisions or what?
Yeah, so we looked at ribosome occupancy. We looked at sort of decoding rates, codon optimality, GC content. What else did we look at? Delta G, overall delta G, like microstructures. And we found that all of them have an impact to some extent. It's about finding the right combination of those factors. Generally, the rule of thumb, I think, sort of follows what we see with mRNA in some aspects. For example, higher GC tends to lead to more stability. Faster decoding is not good, generally. It's a little bit maybe counterintuitive, but it reduces your protein expression probably through compromising stability. So I think, the rules are kind of similar. The other specific things that we did with respect to our codon optimization process was to try to enhance circularization by bringing the termini together through the folding of the coding sequence. I think that's sort of the unique aspect, one of the unique aspects of our algorithm, and we have found that actually helps manufacturability and reliability a lot.
Oh, no, it's just, so we always talk about that circular RNAs are more stable, right? But have your group, so we see papers here and there testing stability of circRNAs at room temperature, encapsulated or naked RNA, but from your experience, have you guys done a systematic analysis, like how long does it really last? Like say at room temperature or in -80?
Yeah, so at -80, it lasts basically forever. So I've seen circular RNA that was thawed after like 2 years, and it maybe had like 10% degradation, 10% single mix, that is. So it was still pretty much intact after a couple of years. Once you get to room temperature, the degradation accelerates quite a bit. But it'll still take probably 30 days for half of it to degrade.
And there's no issue with the translatability of it too?
Well, when it degrades...
at -80, like the...
if you store it at -80, it doesn't prevent iris function or anything like that, from what I've seen. The degradation will reduce the potency, so some of those molecules will basically not be active because they'll be broken. If you leave it out at room temperature for like a month, then I would probably expect that the translation will be a bit lower. Yeah.
All right, I want to thank Dr. Wesselhoff again for an amazing talk and for a vibrant discussion, and we'll sign off. Again, you're at the cutting edge of RNA therapeutics. I'm Christopher Brandon, joined by Daniel Kiss, and thanks for joining.